So we are going to talk about the eye. And so the eye is contained within the orbital cavities of the skull. And here we see our eye, and the white part of the eye that we're seeing, this is our outer fibrous tunic. I'll talk about specific structures in just a second. But attached to the outside of the eye are several of these skeletal muscles called extrinsic eye muscles. And these all control voluntary movement of the eyes. Now, although our eyes can move involuntarily, they are skeletal muscles because we can move our eyes if we choose to. And they're under control of, or the impulses would be sent along neurons contained within the ocular motor, abducens, and trochlear nerves. These are three cranial nerves that you have to be able to identify, and they are all involved in eye movement. And it's through these extrinsic eye muscles that are attached to the outside. Now, for extra credit on the exam, so for our purposes, you just need to be able to tell me that these are extrinsic eye muscles, but if you want bonus, you should be able to identify the specific extrinsic eye muscles. So inferior oblique, because it comes off at an angle, or inferior rectus, because it's a straight muscle, lateral rectus, because it's on the lateral side, superior oblique, superior rectus, and medial rectus, okay? So if you can identify the specific extrinsic eye muscles, you'll get bonus. So with the eye, there are three major tunics meaning layers, and there's an outer fibrous tunic, a middle vascular tunic, and an inner nervous tunic. Okay? And so this outer fibrous tunic is made up of two major components. The anteriormost, which is right here, this is the cornea. This is the cornea. It's kind of bubbled out. And the cornea is primarily a fibrous connective tissue, abundant collagen fibers, but it's transparent. Unlike the remainder of the outer fibrous tunic, which is what you think of as the white of your eye, which is continues posteriorly all the way back until it merges with the dura mater that surrounds. This would be the optic nerve coming off the posterior side of the eye. And so all of this white stuff on the very outside, this is called the sclera. And the sclera is dense fibrous connective tissue with abundant collagen fibers. But the sclera is opaque. It's not transparent because the collagen fibers are in an irregular fashion, so they're randomly or somewhat randomly arranged, whereas the collagen fibers in the cornea are so regularly arranged that they are able to be transparent. And that's a good thing because this transparent cornea allows to focus light rays, and it allows those light rays to enter the eye and will ultimately go through this pupil or this opening, but this transparent cornea allows for light rays to actually enter, and it focuses it to go through the pupil and enter through the lens to get to the nervous tunic. Okay, so that's the cornea, which is the transparent fibrous portion, anteriormost, and then the remainder white portion, which will then be continuous with the dura mater that surrounds the optic nerve, that's the sclera. Now the middle vascular tunic, exactly what it sounds like, it's going to be very highly vascularized. And when you have the models in the lab, if you open up the eye and take off the sclera and the cornea, and you take out what's left inside, you'll see this dark, kind of brown, purplish layer with abundant blood vessels. That would be the choroid, and that's a major structure of the vascular tunic. Vascular tunic because it supplies blood vessels for nutrients. And when you take off the sclera and the cornea, and you see that dark brown structure, the choroid, right? It's dark brown because it has abundant melanin. Melanin is a pigment which absorbs light, and so this dark choroid will absorb any extraneous or excess light and keep the inside of the eye relatively dark. So when light rays do enter, they can, only what you are looking at will be able to stimulate the photoreceptors. So that's the choroid. Now the ciliary body, so we're looking at a lateral view of this eye. So this middle layer, this would all be choroid, okay? And when it extends anteriorly, this portion this portion right here, anteriormost, this is the ciliary body. And let me see if I have This is looking at from a superior point of view. Okay, so we've cut the eye and we're looking at a superior view. So here these would be the ciliary bodies, right, which is made up of smooth muscle fibers. And then the ciliary processes, which attach to the ciliary processes, are these structures right here, which are the suspensory ligaments. And these suspensory ligaments hold this lens in place, okay? So, and I'll show you all in lab what the ciliary body and the ciliary processes are. Now on the list, I have suspensory ligaments listed. You are going to want to cross off suspensory ligaments and add ciliary processes, because you'll be able to see the ciliary processes on our models, but not the suspensory ligaments. So let's talk about the structures of these, or the functions of these structures. So the ciliary body, which is made up of ciliary muscles, those are smooth muscle fibers, and the ciliary processes, and attached to those ciliary processes are these suspensory ligaments that exactly what they sound like. So they suspend the lens in place, okay? And the lens helps focus incoming light rays to direct them to the inner nervous tunic so that light can stimulate the photoreceptors. So when you are looking at a distant object, and then you switch to looking at an object that's much closer, what happens is your lens has to accommodate, right, to focus the light differently. And so the ciliary muscles will contract or relax, and pulling on the ciliary processes, therefore pulling on or relaxing the suspensory ligaments. And so it changes the shape of the lens to allow for you to focus light when you're looking at a distant object or an object up close. So the cool thing about the lens is the lens actually develops from epithelial cells, and these epithelial cells will lack organelles, and they lack organelles because organelles will scatter light, right? So they lack organelles, and they have abundant proteins called crystallins. And so crystallins and the fact that they lack organelles allows for the lens to be transparent, okay? So I added a few pictures in here to help me, and I'll post the updated PowerPoint. I'm going to go ahead and switch to this image right here, which shows you, here's our ciliary body with the ciliary processes. These structures right here, which are suspending the lens, these would be suspensory ligaments, okay? And what happens is, normally, if we're looking at a distant object, the ciliary body will be relaxed, and what happens is these suspensory ligaments have tension in them, and so it pulls on this lens in both directions, and the lens is somewhat flattened. This is when you're looking at a distant object. Well, when you look at an object, when you switch and you look at an object up close, what happens is that we need to relax these suspensory ligaments so that the lens can become a little bit fatter, a little bit more round, to focus the light differently so that we can look at the up close object. And so the ciliary body will actually contract, and that relaxes these suspensory ligaments, allowing for the lens to somewhat bulge out. And this process is called accommodation, okay? So another structure, the iris and the pupil, and I actually need to look at an anterior view right here. So this, when you look through the cornea and you see the colored part of your eye, when you say I have blue eyes, I have brown eyes, I have green eyes, what you're actually talking about is the pigment of the iris. So this structure right here, right, this is the iris. And the iris is, think of it as like a diaphragm on the microscope. And it actually, the iris is made up of radial and circular smooth muscle fibers, which allows that when they contract and relax, they will change the diameter of this pupil. And the pupil is literally just a hole, and it's a hole that allows for light. When the light passes through the cornea, then it will pass through this pupil to enter into the posterior cavity of the eye, okay? And so this is the iris, and the iris can contract and relax and change the diameter of the pupil, or the hole, that allows a certain amount of light through. And if you've ever looked in a mirror, right, in the bathroom, and you turn the light off, and you stare at yourself in the mirror, and then you flip the light on really fast, and you look at your pupils, you'll notice that when you flip the light on, your pupils hurry up and shrink in diameter. The diameter gets smaller. It decreases in diameter. That's because now all of a sudden abundant light is available. So when there is a lot of light available, the pupils will be very small, right? Now, if there's dim light or very little light available, then what happens is this pupil will get larger in diameter to try to allow as much light as possible into the eye so that you can see, okay? So that's the iris and the pupil. And so those are all the structures of the middle vascular tunic that you should be able to identify. Now, the inner nervous tunic is made up of the retina. And let me go back to this one right here. When you're looking at the inside of the eye, the lining, the majority of the posterior and sides of the inner eye, all this red, kind of pinkish red stuff, this is all the retina, okay? And the retina contains all of those photoreceptors that are able to respond to light and send impulses to the brain, okay? So this is actually a picture of a model that we have in lab. All of this pinkish stuff on the posterior or the back of the eye, this is the retina, okay? And there are a few structures within the retina. So photoreceptors that you find in the retina are rods and cones. Rods are responsible for vision in dim light, so they really just kind of sense motion, and it's black and white vision. Okay, so they're really sensitive to light, and they just give black and white vision and motion detection. Now, cones have the ability to detect color, and they give you your sharpest vision, okay, visual acuity. This structure right here, this is called the macula lutea, and contained within the macula lutea is a really dense, it's densely occupied with lots and lots of rods and cones, okay? So it has a very high density of photoreceptors in the macula lutea. Now this little yellow spot within the macula lutea is called the fovea centralis, and the fovea centralis has only cones. So when light hits directly on this fovea centralis, this is your highest visual acuity. Now this structure right here, which is somewhat white, is the optic disc, and I'm going to show you this image right here. So here, would be the retina. Light comes in and actually has to travel all the way back to stimulate these rods and cones, and then information is sent from these rods and cones to these bipolar neurons, which, remember, you find bipolar neurons in special senses like the eyes. And then these bipolar neurons sends information to these ganglion cells, which then their axons leave the eye through the optic nerve. Well, to exit the eye through the optic nerve, they have to all pass through this area. This is the optic disc. So if you were to flip this structure over and look at the posterior side, you would see the optic nerve exiting right where this optic disc is. And so because this optic disc, this is where all of those neurons are, those axons of the neurons are leaving the eye to go to the brain, there are no photoreceptors in this area. It's all axons of the neurons leaving. And so this is called your blind spot. This is the optic disc. Blind spot because there are no photoreceptors. If the light hits this area, you won't see anything. Let's see, what else do I...